Welcome all. We continue with our epidemiology module, and today in the study design, we'll discuss about the clinical trials. Okay, mainly we'll focus on the randomized control trials. Okay, so RCT is actually a type of cohort study where the investigator decides the exposure. So this is the main thing here, because it is a experimental study. There is a exposure which is assigned by the investigator. So in cohort, what happens? The exposure is decided in the natural course only. The investigator will not assign. So it's actually similar to the cohort study, but assignment of the exposure is decided by the investigator. So in cohort study, nature decides who is exposed, while in RCT, the investigator randomly allocates the exposure. So this random allocation is very important. Okay. So what exactly we are going to discuss today? We'll define the clinical trial and randomized clinical trial, randomized control trial, describe the phases of the clinical trial. This is also SBA. Then we'll understand the characteristic of RCT, and most important, why randomization elements the confounding. Okay, so this is very important. The effect of confounder can be eliminated because of the randomization. Then we'll discuss the strength and limitations of RCT. We'll differentiate cohort studies from RCT and understand ethical and practical issues in conducting RCT. And we'll also solve few SBAs today. Okay, so what exactly the clinical trial is? So clinical trial is an experimental study in which the investigator assigns an intervention to participants and evaluates its effect on one or more outcome. Okay, so it's an experimental study design in which the investigator is assigning the intervention to participant and evaluate its effect on one or more outcomes. So what is randomized control trial? A randomized control trial is an experimental epidemiological study in which participants are randomly allocated to intervention or the control groups, and the outcomes are compared prospectively. Okay, so in a randomized control trial, they are allocated to either group in a random way. Okay, so in a cohort study, nature decides the exposure, while clinical trial, researcher decides the exposure. Okay, so what is the basic structure of RCT? We have some eligible participants, then there will be random allocation by the researcher, and they will be allocated to either intervention group, that is either drug, or they will be exposed to certain intervention, or to the control group where the placebo or the standard care will be given. Remember, it is the standard care. They cannot be left without treatment. Okay, so you need to give standard care, and they will be followed up for the period, follow-up period, and we will compare the outcome. Here also they are asking the questions about the phases of the clinical trial. Okay, so practically there are 4 phases: phase 1, 2, 3, 4. Before phase 1, there is another preclinical phase which actually done in the lab in animals. Okay, 10 to 15 participants, and it is exploratory microdosing is done in the phase 0. Okay, they are also asking the number of participants in the phase or the purpose of that particular phase. What exactly is the purpose of the phase? That is asked in the SBA. Okay, so phase 1, the purpose is to check the safety, toxicity, and dosage of the drug. The participants are less in number, 20 to 100 healthy volunteers. Okay, these are healthy volunteers, 20 to 100. Okay, then in phase 2, we have 100 to 300 patients. Here what we check is the efficacy and side effects. Okay, so safety, toxicity, and dosage in the phase 1, while efficacy and side effects in the phase 2. Phase 1 is on healthy volunteers to 20 to 100, while phase 2 is on 100 to 300 patients. Okay, phase 3 is more number of patients, up to 1000 to 3000 patients, and it will compare the drug with the standard treatment. So whatever actually most of the time trials are carried out, they are phase 3 trial. Okay, phase 3. Before that, the phase 1 and phase 2 are over, and phase 4 is a post-marketing surveillance. Okay, PMS, post-marketing surveillance, and here the long-term safety and rare adverse effects are noted. Okay, so these are the phases. Phase 1 looks for the safety, phase 2 is for efficacy, phase 3 is for comparison, and phase 4 is for surveillance. So safety, effectiveness, comparison, and monitor. Phase 3, we look for the compare with the standard drug or standard treatment. Okay, so why RCT is similar to cohort study? In cohort study, there is an exposure and the cases are followed up for the outcome. Same thing what we do. Only thing is that here the exposure is assigned by the researcher, then the cases are followed up for the outcome. The only difference in the cohort study is exposure is happening naturally, while in RCT, exposure is assigned by the investigator. Okay, so this is the same thing is shown in the diagrammatic way. So there is a randomization of the participants to the either drug or the control group and look for the outcome. So what are the characteristics of the randomized control trial? So it is an experimental study design. Investigator will assign the exposure. Participants are randomly allocated, usually prospective. Comparison between intervention and control group is there. Incidence can be calculated. Relative risk or risk ratio can be calculated. And it is a base design for evaluating the treatment efficacy. Okay. So what are the things which we measure in the RCT? Okay. Incidence can be calculated. Relative risk can be calculated. Risk difference, attributable risk can be calculated. Odds ratio is also possible, but not usually preferred in this clinical trial. But mainly incidence you get, relative risk you get, attributable risk also you get here. Okay, and risk difference also you can calculate from the clinical trial. Okay, so RCT and cohort study both primarily calculate relative risk. Case control study primarily calculates odds ratio. Cross-sectional study commonly uses the prevalence and the odds ratio. Okay, so these things you have to remember: case control, odds, cross-sectional, prevalence, and RCT and cohort study will give you the incidence as well as relative risk also. Okay, so why is randomization important? Okay, so randomization distributes all baseline characteristics equally between the groups. Okay, you may have 2 groups, 3 groups, whatever, but this randomization will distribute all the baseline characteristics equally between the groups. So this minimizes the bias, and more importantly, randomization removes the confounding. Okay, so randomization, this can also be asked in the SBA. How will you remove the confounding? So it is the randomization that removes the confounding. So what is a confounder? Confounder is a third variable that falsely alters the observed association between the exposure and outcome, because it is associated with the exposure also and independently associated with the outcome also. Okay, so there are certain conditions for the confounding. A variable acts as a confounder only if it satisfies all 3 conditions. Okay, it should be associated with the exposure, it should be independently causes the outcome, and is not on the causal pathway. Okay, it should not be on the causal pathway. That means it should not be intermediate between the exposure and the outcome. Okay, so this should be an different entity which is directly linked to the outcome also. It should not be exposure leading to outcome through one factor. Okay, then that cannot be considered as a confounding factor. Okay, so confounder has to be independently causing the outcome also. Okay, so this is the smoking causes the lung cancer. Even the coffee drinking is also here the confounder. Okay, coffee drinking appears to be associated with the lung cancer because smoking is the confounding variable here. Okay, smoking is associated with the both. Okay, so when we want to establish the coffee drinking and lung cancer, smoking can be a confounder. Okay, so how does randomization remove the confounding? Suppose we are randomly assigning 1000 participants. Okay, so we have 1000 participants, then we carry out the randomization. They will be randomized to the intervention group and the control group. So when we do this randomization, then age, sex, smoking, alcohol, BMI, diabetes, genetics, known factors, unknown factors, they will be equally distributed between the groups, because no variable becomes preferentially associated with the exposure. There is no preferential allocation of these particular factors to either of this group. So all confounders will be balanced. Okay, including known, unknown, measured and unmeasured confounders will get balanced. Okay, so randomization is the only study design feature that controls for known, unknown, measured and unmeasured confounders. Okay, so why RCTs are called gold standard? Because there is a randomization, it's a prospective design, it minimizes the confounding, and there is a clear temporal relationship that can be calculated, and it's a strong there is a strong internal validity. Okay, so what are the strengths of the RCT? So it eliminates the confounding. That is the one of the great advantage. Then strongest evidence for causality. because there is a temporal relationship that can be defined. Exposure occurs before the outcome, can calculate incidence, new cases developing during the follow-up can be measured, then relative risk can be calculated. Most clinically useful measure of association is a relative risk that can be calculated, and there is a high internal validity, bias is minimized. Okay, so advantages: there is a randomization, elimination of the confounding, strong evidence for causality, prospectiveness is there, incidence is measured, relative risk will be measured, and there is a high internal validity. While what are the limitations? It is very expensive. Large trials may cost millions of dollars. Then there are certain ethical limitations. Some exposures cannot be assigned. Okay, so you cannot make the participants to get exposed to the smoke or make them smoke. Okay, so there are ethical limitations. Okay, like smoking, radiation exposure, environmental pollution, harmful chemicals. That type of intervention cannot be assigned. Okay, so like smoking leading to the lung cancer, we cannot put the participant at the risk. We cannot make them to smoke or expose them to the smoke. Okay, intentionally exposing the participants to a harmful exposure is ethically unacceptable. Okay, then there is a limited generalizability in the clinical trials. Suppose people volunteering for trials may differ from the general population. Okay, for example, a physical activity trial may recruit mostly retired individuals. Suppose if we carry out some trial for physical activity, in that particular, you may likely to get more of the elderly individuals because they are retired people, they have got a time. So you may get the people from that particular class. While if your study is pertaining, if you want to extrapolate your reports to the young population, when the young population is not there in the study, then that is not a valid. So there are certain limitations about the generalizability. So if you carry out such type of study, it will be applicable to the study population, similar study population. So can results be generalized to young working adults? In this case, we will not be able to generalize it. Okay, then there is a restrictive inclusion criteria, because most of the trials will exclude the morbidity such as diabetes, cancer, multiple comorbidities will be reduced. But practically, when the drug comes into the market, patients may have the associated diabetes, associated cancer, or other comorbidities, and the disease for which the drug is tried, then whether to give or not to give. So there are restrictive inclusion criteria when you carry out the clinical trial. Then exposure must be precisely defined, like physical activity intervention. Should participant walk, run, do yoga, lift weights, etc.? How often, for how many weeks, at what intensity? Everything should be defined. So poor exposure definition may lead to the false negative result. Okay, and there is an internal validity versus external validity. Internal validity deals with accuracy within the study, and it is excellent in the RCT, while external validity is ability to generalize. Sometimes it is limited. Okay, so trials should always be registered in a appropriate registry. So before starting the first participant recruitment, the trial should be registered. RCT protocol should be registered with the clinicaltrials.gov, or clinical trial registries are there. So registration is very important. So why registration? It will maintain the transparency, prevent selective reporting, and prevent publication bias. Ethical accountability will be there. There is about role of FDA and RCTs. The Food and Drug Administration generally requires multiple well-controlled RCTs before approving new drugs, vaccines, and medical devices. Okay, so we'll compare the clinical trial with the cohort study. So cohort study is an observational study, while RCT is an experimental. Okay, so exposure is assigned by the nature in the cohort study, while here in RCT, it is the investigator who assigns the exposure. Randomization is not done in the cohort study, while RCT will have the randomization. Confounding is possible with a cohort, but it is minimized in the RCT. Both will give you incidence and the relative risk. Both are costly. Okay. Then ethical issues are less with the cohort, but they will be more with the RCTs. And internal validity is moderate with the cohort, while excellent with the RCT. Okay. So from examination point of view, RCT is an experimental study design. Investigator is assigning the exposure. Randomization removes the confounding. Incidence and RR can be calculated. Gold standard for evaluating the intervention. Then smoking cannot be randomized because of the ethical concerns, and RCT often have limited external validity. External clinical trials should be prospectively registered. Okay. RCT stands for in mnemonic: R for randomization, C for controls confounding, and T for temporal relationship can be established. Okay, so that finishes the topic of clinical trials. We have few SBS. Doctor, can you please read the SBS?
Yes, sir. A researcher randomly allocates hypertensive patients to receive either drug A or placebo and follows them for 5 years to compare the stroke incidence. The study design is case control, cross-sectional study, cohort study, randomized controlled trial. Sir, option D, randomized controlled trial.
And here, most important is a researcher randomly allocates. Okay. So look for such kind of words because they are related to the concept, the definition as such. The investigator determines the exposure status through random allocation. Next.
The greatest advantage of randomization is reduces cost, increases sample size, elimination of confounding, better external validity. Sir, option C, elimination of confounding.
Correct. Okay. So randomization equally distributes both known and unknown factors. Okay, confounders. Next.
Which of the following exposures cannot ethically be studied using an RCT? Aspirin therapy, calcium supplementation, smoking, influenza vaccination. Which of the following exposures cannot be ethically studied? Sir, option C, smoking.
Yes. So assigning participants to smoke would intentionally expose them to the harm. Okay. So there are ethical principles. Okay. So there will be violation of the basic principles of ethics. Okay. Do no harm. Next.
Yes. Which measure of association is primarily obtained from an RCT? Odds ratio, relative risk, hazard ratio only, standard mortality ratio. Sir, option B, relative risk.
Option B, relative risk. Next.
A major limitation of RCT is cannot calculate incidence, cannot establish temporality, ethical constraints always affected by confounding. Sir, option C, ethical constraints.
Yes. So just to summarize, randomized control trial is an experimental prospective study in which the investigator randomly assigns exposure, thereby eliminating confounding and providing the strongest evidence for the causality. Okay, so that finishes the today's topic of study designs, especially the clinical trials. Thank you.